Hello and welcome everyone to the Lexogen and BlueBee webinar. I'm Ekaterina Alexeyeva, a Senior Marketing Manager at Lexogen, and I will be moderating this webinar today. Before we start, I have some announcements. Please note that all attendees are in listen-only mode. Our webinar today is about user-friendly data analysis for cost-efficient and streamlined RNA sequencing. This webinar will take about 40 minutes, including 25 minutes of a presentation and 15 minutes of the questions and answers. Our speaker today is Lauren Shehan, the US application scientist from Lexogen. For the Q&A, she will be joined by Jorge Zapeda and Falco Hoffman, who are also application scientists at Lexogen, and Andreas Klingenhoff, who is a project manager at BlueBee. It's my great pleasure to turn it over to Lauren. Lauren, welcome, and please go ahead.
So finally, I will be discussing the updates made to Lexogen's data analysis tools on BlueBee and how they will help enhance user experiences. So as mentioned before, if you do have any questions during this webinar, please go ahead and type them, and myself, along with my fellow panelists, will help answer them. And if you have any questions come up after this webinar, please feel free to email us at support@lexogen.com. So Lexogen is a biotech company focused on enabling analysis of the complete transcriptome. Lexogen was founded in 2007 and is based in Vienna Biocenter in Vienna, Austria. We also have a subsidiary in New Hampshire, USA, which was founded in 2014. Our company is comprised of a multinational team and a worldwide distributor network. And this allows access to researchers across the globe to our products and services. So Lexogen offers an extensive portfolio of products to enable sequencing of the transcriptome. Although this webinar pertains specifically to tools for data analysis, I want our listeners to be aware of the end-to-end workflows that we provide. So here you will see SLAM-Seq, which I will be getting into later. So SLAM-Seq is a method for measurement of newly synthesized or existing RNA in cultured cells. It enables the resolution of RNA synthesis and degradation kinetics. Our SLAM-Seq portfolio, as seen here, includes an Explorer and Kinetics module, which covers the entire metabolic RNA labeling experiment, from optimizing labeling conditions to labeling nascent or existing RNA and alkylation for downstream NGS library preparation. Total RNA for SLAM-Seq experiments can be used directly with Quantic, which is Lexogen's mRNA-Seq gene expression analysis library prep kit. And we also have a data analysis pipeline specifically for analyzing SLAM-Seq data, which is called SLAM-Dunk. And again, I'll be getting into this later on in the presentation. So here you will see some of the available products that we have for preparing your RNA, as well as generating libraries for sequencing. For RNA preparation, we have Split for the extraction of RNA from a variety of samples. For ribosomal depletion, we have RiboCop, and this is for use in human mouse rat or for use in bacteria. And for our poly-A kit, it is for the rapid and highly specific enrichment of polyadenylated RNAs from total RNA samples. In terms of NGS library preparation, we have 4 main kits depending on the needs and goals of our researchers. So Quantic is our gene expression analysis kit, and I'll go into this product in upcoming slides. For whole transcriptome sequencing, we have both Coral as well as Sense. And finally, our small RNA-Seq kit allows for the generation of small RNA libraries for Illumina sequencing directly from total RNA or enriched small RNAs. Also listed here is our product for full-length cDNA preparation called Teloprime, as well as our spike-in RNA controls called SERVs. If you want more information on these products, please feel free to contact us or visit our website. Finally, the part of the portfolio that we will be discussing today is our analysis tools for NGS data. On the BlueBee Genomics Platform, we have pipelines for analyzing data from Quantic, Coral, and SLAM-Seq experiments. I will be going into each one of these later on in the presentation. On the bottom of this picture, you will see that Lexogen also offers in-house services for the preparation, sequencing, and data analysis of your RNA samples, Again, please visit our website or contact us if you want more information on any of these products or services that I've mentioned. So now a little bit about BlueBee. So BlueBee is a genomics data analysis platform that Lexogen has partnered with to allow for the availability of easy-to-use data analysis tools. BlueBee has deployed Lexogen validated analytical pipelines, which deliver production-ready, robust infrastructure that is regulatory compliant and easy to use for any researcher. So with BlueBee, you can have the confidence that your sequencing data will be both productive and reproducible. Lexogen's pipelines on BlueBee, especially with our new web solutions, are easy to use and no prior bioinformatic experience is needed. In just a few clicks, you can go from uploading your FASTQ files to downloading your analyzed data. Additionally, you can save both time and money by performing your own analyses. Most importantly, know that BlueBee offers a secure and compliant environment to support both research and clinical applications at any scale. So Lexogen and BlueBee, we began collaborating back in 2016 with the launch of our pipeline for Quantic data analysis. And still true today, all of our Quantic kits do come with complementary BlueBee activation codes, which allows users the access to Quantic data analysis pipeline for free. So this pipeline has been further developed since 2016 to include differential expression analysis, or DE analysis. And over the years, we have also developed and implemented pipelines for several other of our products, including Coral and SlamSeq. And today, we are happy to announce the release of our new user interface, which I'll take you through later in this presentation. And moving forward with our collaboration, we will continue to develop and implement solutions to allow for the ultimate user experience. So up until now, I want to go into a little of opportunities and challenges in transcriptomics. So whole transcriptome sequencing, be it mRNA or total RNA-Seq, is what has been used to look at de novo assembly of transcripts, detect isoforms, and perform expression profiling. With this technique, you start with multiple fragments per transcript and end with fairly involved bioinformatics for the assembly and analysis, which results in a lot of time spent and high cost to obtain your NGS sequencing results. However, for expression profiling specifically, it would be much easier if you could build a library around the fragment of interest and then sequence just one fragment per transcript, as seen here on the right. Now you begin without the need to fragment and end with a much simpler bioinformatic approach, essentially counting the number of sequences you have. So this is exactly what you do with Lexogen's Quantic library prep kit, and now we'll discuss a little bit about this. So compared to other RNA-Seq library preparation kits, Quantic provides many benefits. It provides cost-efficient expression profiling, where researchers have the opportunity to multiplex up to 9,216 samples and save sequencing space. Our kit provides options for low inputs, going down to 100 picograms of input material. It can also be utilized for heavily degraded or FFPE samples. There are also options available for targeted RNA-Seq, where researchers can design their own primers for targeted specific transcripts. Most importantly, we provide an end-to-end workflow with complementary and simple data analysis through BlueBee. And Quantic over the years has become increasingly popular because of these benefits. And today, Quantic has been featured in over 440 publications. So Quantic data analysis on BlueBee is extremely user-friendly and can be used by any customer. Again, no prior bioinformatic knowledge is required. When you buy a Quantic kit, you will get an access code that is used on BlueBee's site for analysis at no cost to you. We also have a promotion going until the end of August where extra Quantic codes of up to 1.4 or 1.5 gigabytes of input data are offered free of charge. With just a few clicks, you can upload your data and perform analyses. And again, BlueBee offers a highly secure platform with a variety of reference genomes available. And if your species is not listed, you can always contact us at support@lexogen.com and request that a species be uploaded for a fee. So to analyze your data on BlueBee, there are 5 simple steps to get started. First, registration using your activation code found on your Quantic kit, which I'll direct you to later in this presentation. Next, connect your sequencer with the BlueBee service connector or a cloud-based connector such as Amazon, BaseSpace, or Google. FASTQ files can then be uploaded via the BlueBee service connector, a cloud-based connector, or our new feature, the drag and drop option. Following this, you would choose your pipeline based on the kit you have purchased. And then lastly, you can view and download the output files, which include mapping stats, read counts, and FASTQ files. Additionally, you can also, for Quantic, run DE analysis to obtain a list of differentially expressed genes, heat maps, PCA plots, and MA plots. So when you receive your Quantic kit, you can find your complementary BlueBee activation code on the inner kit box as shown in this picture here. Please note, if you use a kit with a unique molecular identifier, abbreviated UMI, utilize the Quantic forward UMI pipeline. Running the forward pipeline on UMI libraries or vice versa will result in failure, so I just want all of our listeners to be aware of this. So here's an example of the workflow of the Quantic pipeline as well as the DE analysis pipeline. Our pipelines include published open source tools, which are prefigured and for non-experts. As mentioned previously, output files include mapped reads, gene expression tables, MA and PCA plots. And all of our pipeline workflows, which are pictured here, can be found in their respective user guides. So with Quantic, you only sequence 10% of what you would normally sequence. So this allows for a high level of multiplexing. This high level of multiplexing will dramatically lower the cost of expression analysis per sample. So as seen here, if you include the cost of sequencing, analysis, and library prep, the cost per sample can be as low as $36. So now getting into what's new with our pipeline and also the environment. So overall, these implementations have resulted in enhanced user experience. There are now more ways to upload and download your data and several new features such as the Integrative Genomics Viewer, IGV, a support tab for submitting questions and also FAQs. This new environment is also very interactive and very easy to navigate. On the right here, you'll see two of our landing pages along with the URLs. On the top, this showcases the available pipelines for analyzing Quantic, Coral, or SlamSeq data. And on the bottom, this is an example of the landing page for a specific pipeline. So in this case, it's Quantic. So now there's more options for getting connected and also uploading your data. In addition to the BlueBee service connector, you can now upload data through cloud connectors such as BaseSpace, Google, and Amazon. Instructions on how to utilize these cloud connectors can be found in our FAQs and will soon be available in our user guides. Additionally, you can drag and drop files directly from your computer to your browser. So with the Integrative Genome Viewer tool, you are now able to visually explore large integrated genomic sets within the environment itself, as depicted here. We have included an FAQ section, which can be found on the top right and accessed by clicking the question button. And here in this picture, you'll see that this button is circled or in a red circle. Here you will find information about available species, where to find your activation code, and also how to interpret your data. So if you have any questions or run into any problems when analyzing your data on BlueBee, you can now submit a ticket or a question, which will be directed to our technical support team at Lexogen. You can generate this ticket by clicking on the support button on the top, and again, it's circled here in red. And alternatively, you can reach out to us with any questions you have at support@lexogen.com. So that wraps up our new features, but I wanted to also briefly touch on the other two kits, which we have data analysis pipelines available on BlueBee. So Slam Dunk, as mentioned before, so this was developed by a multi-group collaboration. and is a T to C aware alignment software package based on NextGen Map 49, developed to recover TC conversions from SlamSeq datasets. So SlamDunk is not included with the SlamSeq kits, but available separately. The code to access automated data analysis on the BlueBee platform is provided upon purchase at Lexogen. So input requirements are the same as Quantic, so it's FASTQ files. And output files include T to C conversion rates and a comprehensive statistics for each of the 3' UTRs that you have. So similar to Quantic, we have an outline of the SlamDunk pipeline in our user guide. Overall, this pipeline is very fast, efficient, and affordable for processing and interpreting your SlamDunk data. It is currently available again for a fee. So please visit our website for more information or contact us at support@lexogen.com. So our final kit I'll be discussing is Coral. So Coral is our kit for whole transcriptome analysis, and it enables the fast and cost-efficient generation of unique molecular identifiers, UMIs, labeled stranded libraries for the analysis on Illumina NGS platforms. So for analyzing Coral data, we have a pipeline that is straightforward and easy to utilize. As you can see in the picture to the right, this pipeline includes UMI deduplication and generates several output files such as gene and transcript abundance and multi-QC. And Lexogen software Mix2 is used for data analysis within this pipeline. Mix2 is precise in determining transcript abundance and has exceptional reproducibility rates. Additionally, runtimes with the software is fast and leaves a small memory footprint. Oh, and as mentioned previously, we do have a promotion going on until the end of August where complimentary BlueBee codes will be included with our Coral kits. So finally, I wanted to touch on reference genomes available and how to request additional ones to be added. So for Quantic and Coral, both of these pipelines include 48 publicly available reference genomes, and this now includes SARS-CoV-2 genome. So for a full list of the reference genomes available, you can either visit our website under our FAQs, or you can reach out to us directly. For SlamDunk, we currently have human and mouse reference genomes. Again, if you want to add any species to this list, you can go ahead and contact us at support@lexogen.com. So to summarize what Lexogen and BlueBee have available, we currently have three environments. Environments available for the following kits. So Coral, our whole transcriptome analysis library preparation kit. We have Quantic, our gene expression analysis library preparation kit, and SlamSeq for metabolic labeling of RNA. Again, SlamSeq experiments can be analyzed using SlamDunk pipeline on BlueBee. So with that, I would like to thank you for your attention, and I would like to again introduce our panelists who will be helping answer questions. So Jorge is an application scientist at Lexogen located in Vienna, and Falco is also an application scientist at Lexogen who specializes in bioinformatics. Then we also have Andreas from BlueBee. He is a project manager. Again, if you have any questions, please type them into the chat window, or if after this webinar you have a question, contact us at support@lexogen.com. Thank you.
Thank you, Lauren, for this great talk. Please, as Lauren mentioned, you can submit your questions in the questions box, which you can find on the right side of the webinar platform. We have around 15 minutes and we will try to answer as many questions as possible. We have some questions which were submitted during the registration process. So we will start with those questions. I saw also some questions were already answered during the presentation itself, so we will focus on those which were not touched. All right, so our first question, does this platform analyze data from dicot plants?
So right now, we currently do not have dicot plans on our reference genome list, but this can be implemented. So again, like I said, if you want to upload or have a reference genome added, you can contact us at support@lexogen.com.
Thank you, Lauren. And another question, is your RNA-Seq analysis pipeline open sourced?
So I can go ahead and take this one too. So it is implemented using well-established open source tools, and a more detailed description is available in each of our user guides and also on the Quantic product page itself.
All right. I'm involved in volatile mediated transcriptional changes in rice. Can you please suggest an appropriate protocol?
Yes, so rice is available as a reference genome, and it's available for both Quantic and Coral pipelines. So depending on a protocol, it depends on your experimental goals and what you want to know. If you wanted to analyze the complete transcriptome of your samples, I would recommend going with Coral. If you're interested in only the gene expression differences, then I would say Quantic would be the product for you.
Mm-hmm. Thank you, Lauren. How long is the data stored on BlueBee?
The results of the analysis are stored for up to 1 year in your account. You can, of course, delete data at an earlier time, download it to your local infrastructure, but it is kept available in your account for 1 year.
Thank you. Another question, is it possible to share data with other researchers in other regions?
What you can do is you can invite other users from all over the world to your account, give them access to your analysis, to your data. They have full functionality in there, and you can collaborate on the data in your account in a secure environment.
Thank you, Andreas. And how do I know that my data is secure?
Security is one of the main focus at BlueBee. We put quite some effort in that. Data is encrypted in flight, in rest all the time. We are ISO certified. All our partners we work together with for data processing are ISO certified. That is really one of the main focuses at BlueBee.
Thank you, Andreas. Out of interest to understand the technology a bit better, could you explain a bit more detail how your approach achieves higher multiplexing and thus lower sequencing prices? Jorge, maybe you can answer, or Lauren?
Sure. Yeah, so I think this question is about Quantic. So Quantic allows higher multiplexing, more or less 10 times higher multiplexing than doing whole transcriptome analysis, because this approach focuses only on the 3' end of the genes, which is enough for counting, basically, and to quantification of genes at the gene level. And therefore, we have made some calculations, and it's also been published in the literature that with this, one can multiplex more or less 10 times higher, and therefore reduce the sequencing costs.
Thank you. For mRNA-Seq, do I need all 3 kits, split RNA extraction, ribocop ribosomal RNA depletion kit, and poly-A RNA selection kit before library prep?
So I'll take this one. Specifically for Quantic, no, you don't need any of these products before. For the split RNA extraction, yes, if you do not already have an extraction kit for getting RNA from your samples, you can use this. But as far as ribocop for RNA depletion and poly-A, you do not need this for Quantic. However, for Coral, you would need one or the other.
Thank you, Lauren. Can I just send mRNA to your company for mRNA sequencing?
Yes, I'll take this question. Yes, you can definitely send us your RNA, or depending on the sample types, even the cells, and we can also do the RNA extraction, and then we can offer either a whole transcriptome analysis with Coral or Quantic library prep in data analysis as well.
Thank you. Is the recommended minimum target size as number of genes or exons for the Quantic Flex kit?
Yeah. Yes. Well, this really would depend from the sequence itself. So the amplicon, it has to do with the PCR. So it has to do with the sequence content, as well as how well can this be amplified. Regarding to the number of genes or exons to be amplified, well, that it really depends on the experiment. In theory, even just one is possible. But then so you can buy you can buy different kit sizes, and this can even be multiplexed higher. We can also offer primer design for big projects like thousands. For this, you would have to contact support, but in principle, from one gene on, and then you could try the amplification, and we can also provide support for primer design to ensure that your experiment would work.
Thank you, Jorge. Is there a limit to how many replicates or treatment conditions within a single experiment can be analyzed for differential expression using your interface?
So I take this one. So generally, like the differential gene expression testing is being done by DSeq, and this is done pairwise. So you can input, I think there's no clear limit on how much you can input as one group or the other, but at the moment we only support pairwise testing. So if you want to do more complex analysis, as for example, the likelihood ratio testing with DSeq, where you have different treatment and time series or so, this is currently not yet available.
Thank you. Please contact us if you have any requests related to Lexogen or BlueBee solutions, and we will be in touch soon with the recording of this webinar. And I want to thank the speaker, Lauren, and all our panelists today. And also thank you, attendees, for your attention and questions. We will be in touch soon. Goodbye.